But in recent years, we've discovered how to build completely new proteins. And so today I'm going to tell you about the methods we use to design completely new proteins and some of the problems we're trying to solve with those new proteins. So just to remind everybody how the flow of information in biology works, we have genes in our genomes that encode amino acid sequences, and those amino acid sequences fold up into proteins which carry out a biological function. So in protein design, we proceed in reverse. We start with a function that doesn't exist. For example, binding tightly to a cancer target. From that function, we build a protein which we predict would have that function. We work backwards to identify an amino acid sequence and then a DNA sequence which encodes that protein. And then, in a key step, we make the protein and test it in the lab. So everything I'm going to show you today, we started with a function we wanted to make, to create. We designed a completely new protein that we predicted would have that function. And then we made a synthetic gene that encoded the protein, put it into bacteria or yeast, made the protein to see whether it did what we designed it to do. It's inspired by generative methods used to create images. These are diffusion methods where you take a large training set, for example, of images, you noise those images to different extents, and then you train a model to remove the noise. And once this is done, you can start with completely random image and progressively denoise it to generate a new image that looks like it came from your training set, but in fact is completely new. And in the same way, we took all the protein structures in the protein data bank, noised them to different extents, and then trained a model, RF diffusion, to remove that noise. And then once we had done that, we could start with completely random noise, and then this shows a trajectory here. We progressively remove the noise, and we end up with a protein, something that looks like a protein structure. It's not from the protein data bank, it's completely new, but it looks like it could have been. Well, so that is kind of an academic exercise, but it's not that useful. But remember, in the image case, you can condition it. You can say, generate an image of a cat sitting on a table, or anything else you want, and the model will now carry out this generative diffusion process and generate an image with that property. So we can do the same thing with proteins. We can say, generate a protein, for example, that binds a particular target. So this is the insulin receptor, and here you see a diffusion trajectory where it's not occurring in isolation. It's in the context of the insulin receptor, and you can see the protein coming together to form a structure that looks like it is. It could be in the protein structured data bank, something that would bind the insulin receptor. It's complementary in shape, but it actually is a completely new protein. And so today I'm going to tell you about applications of these methods in 3 different areas: in medicine, technology, and sustainability. So in medicine, I'm just going to give you a few examples. Here is one of the components of snake venom, a toxin. And when we carry out the diffusion process, we generate proteins which bind to the components of snake venom, this component, the toxin. And these proteins protect animals from the toxin. So here, animals that don't get this blue protein are completely, cannot survive the toxin, whereas if they get the protein, they do survive. And Susana Vasquez-Torres, who did this work, actually made binders in this way to a number of the different components of toxins, of venoms. And now she's really interested in pushing this out. And currently, antivenoms for snake bite are made by injecting horses with venom and collecting the antisera. As Gaurav mentioned, we've started many companies, and this is sort of an example of how it happens. People design a new molecule, and then they want to get it out in the world. So in inflammation, a major target is the TNF pathway. It's the target of drugs like Humira, for example, and Enbrel. So this is, you see a diffusion trajectory carried out in the presence of the TNF receptor, and we get this very interesting protein which kind of conforms to the surface of the receptor. And this is an experiment where animals that were undergoing an inflammatory response were either given Enbrel or this designed protein. And here the designed protein actually turned out to be quite a bit more effective, as you can see. So we're very excited about this and molecules like it being useful for autoimmune disease. In cancer, we've been making de novo designed versions of immunomodulators like cytokines. So here's a design that brings together the two components of the IL-21 receptor, and our collaborators have found very promising results with this in a pancreatic cancer model, where this protein activates the immune system to fight the cancer much more effectively than the natural cytokine does, probably because it's more stable. The signaling persists for much longer. And we can further condition this diffusion process by specifying not only the target, but the type of protein we want to generate. So in this case, we specified that we wanted to make an antibody-like structure, in this case, a nanobody. And this is the influenza virus. And so what's generated is a protein which binds to the virus, but is also an antibody-like structure. And here's a cryo-EM structure showing the how close the design model comes to the actual structure. So in white is the what we were designing and on the computer, and in blue is what we were trying to make. And we've recently extended this to full and to antibody-like molecules with heavy chains and light chains, single chain SCFEs. So we're this, I think, is a much more straightforward way of making antibodies than how the pharmaceutical industry currently does it. We can also make binders to peptides. For example, here I'm showing a couple peptides, amyloid beta and tau, that form amyloid-like structures when they are allowed to interact in solution. We can design proteins which kind of sandwich the peptide, as shown here, and you can see this is measuring the extent of amyloid formation. This is A beta, and this is tau without, and then with the designed protein added. So they're very effective at suppressing amyloid formation. There's been a lot of controversy about the role of amyloid formation in neurodegenerative disease, but these at least provide sort of tools to more specifically address that question. Okay, so now I'm going to shift gears and show you some applications in technology. So far I've told you about soluble proteins, but we can apply these methods to make transmembrane proteins. Nanopores. So here, these describe, here you see de novo design nanopores with increasing size pores. And as the pores get bigger, the current that can go through those pores increases. And you can see that increase as we go along here. So this is very interesting because we can now make new types of sensors. So for example, we can take a protein that's been designed to bind a small molecule, in this case cholic acid, and put it at the top of the pore. And in the absence of cholic acid, this top part is kind of flickering back and forth. But when we add cholic acid, the pore closes. So this we believe will be a route to a really broad range of new sensors. We can go beyond this by making pores that insert not into biological membranes, but into silicon nitride chips. This is interesting. Now, nature never evolved proteins to insert into silicon nitride, but there's a lot of reasons why that's interesting to do now, because we can pack pores much more densely on silicon nitride than we can on lipid bilayers they're much more robust, and they're obviously more readily couplable to electronic devices. So here are some of the really kind of exotic-looking new proteins that we've designed for this. And here you can see current that is that these proteins will insert into these pores and generate stable current traces. And this has been a great collaboration with Jens Goodluck's lab here at the UW. So we're very excited about this as new ways of making sensors and new sequencing approaches. We can design proteins using very similar methods that bind to DNA for things like gene targeting, for gene repair. So here's a designed protein that's been designed to bind to a particular DNA sequence. Here are the DNA bases, and here's the designed protein with side chains that come out and interact with those bases. And here is experimental data showing the specificity of the design. So we were targeting this particular sequence, TGCACA, and when mutations are made, the binding affinity gets much weaker. The redder it is, the more weaker it is. So this shows that we can make new proteins that are sequence-specific DNA binding proteins.